Hey again, everyone. Thanks for joining us for our third iGEM webinar for 2020. For today, we'll be exploring how to master Benchling Molecular Biology in this extended webinar today. So we're really excited to welcome many of you back again. And for those of you who are joining us for the first time today, I just want to give you some quick introduction. So my name's Johnny Trung. I work here on the academic program here at Benchling. Before working here, I actually finished my PhD in chemical biology at UC Berkeley. In graduate school, I was studying these regulatory RNA elements called riboswitches, really understanding how to engineer them for fluorescent detection. Now I'll actually turn it over to my other co-host to introduce herself.
Hi everyone, my name is Sierra Ray, and I'm a customer success specialist here at Benchling on the customer experience team. Before my time here, I previously conducted research on proteins, specifically on enzymes involved in nucleic acid replication. And before that, I received my master's in applied molecular biology, focusing on microbiology from the University of Massachusetts, Amherst.
Awesome. Thanks for that introduction, Sierra. So I want to give everyone a brief outline of the webinar we have planned today. We're actually going to be starting off with talking about how iGEM teams could be identifying the needs that they have for biology software in a way to better evaluate solutions. Then we'll discuss really how your biology software relates to something important like your iGEM deliverables for this competition. And then Sierra is actually going to summarize an overall workflow for the demo that she has prepared for you all today and go through that. And then to end it all off, we're going to make sure there's a dedicated Q&A session to make sure we answer any specific questions you may have. And let's go over some really quick housekeeping items with the Zoom software we're using today. We really want to make this interactive, and so we plan to have a few quick, short polling questions just to get a sense of some ideas and some topics that you guys have, especially related to these areas that we're going to cover. And then additionally, we're going to have, in addition to this dedicated Q&A session, if you have any questions at any time, feel free to use the Q&A button at the bottom of your screen Zoom panel and send a question there. We'll either answer those questions through the chat, or we'll actually answer them live at the dedicated Q&A session. And just a reminder for some of you all that today's webinar has actually been a part of an ongoing webinar series that we have for iGEM teams. If you missed any of our previous ones, don't worry about it. They're actually available to stream whenever you want on YouTube on demand. Most of the content in this webinar, unfortunately, won't go into detail about those things we covered previously. So we highly recommend you look back through these when you get a chance. So now before I go any further, I actually do have a few questions for you all. So I'm going to actually start by opening a poll and getting some responses from you all. We're really curious about some of the biology design software you're using today, and additionally how you have heard about them. We really want to understand how you think about using new tools and really what spurs you to make those changes. So you'll see there's a series of 2 questions. Feel free to just answer for all of those that really apply to you. So just take some time to read through. I know it's kind of a lofty question, but really, really interested to see what you all think. And then I'm actually going to share these results with you all, just so you can see what some of your other peers are using. All right. So as you can see, a large majority of viewers in terms of using Benchling software are using Benchling software in addition to a lot of other types of biology design. Additionally, it seems like many of you are actually getting the results from your research or your iGEM mentor, which is awesome. Now, I think something I really want to share with you all is my own kind of story where when I was an undergraduate, like many of you are now, I never really questioned the tools that my mentors told me to use when analyzing data and designing constructs. And in many ways, I wish I did and more so. While I was in graduate school, I really learned that identifying really my personal research needs first can often I could often better find more effective solutions that really remove busy work and increase my productivity. So really, I hope that as an iGEM team, I want to introduce you all to a few questions that hopefully you can all answer together and as early as possible. And so first, I really want you all to hopefully explore, what aspect of synthetic biology do you think your team will focus on? And with that, was it going to require a lot of interdisciplinary research? And if so, will that require different tools that you might be using now? Second, think about the design tools you're already using. What are some of the pros and cons that you've seen and that you've observed? And additionally, is it going to be relevant to your iGEM project now, or is it something that you kind of learned how to use, through courses or other through other opportunities? And lastly, will everyone on your team be able to access the software the same way? And will you be able to give that access to other people in the way you want to? Hopefully these guiding questions can be really helpful for you all to think about as a team. And so I want to go through this exercise a little bit more. And so for me, if imagine if you and your team went through this exercise, I'm going to give you an example if I had done it myself. And I'd recommend essentially breaking out maybe all the input from your team into a couple of 5 to 6 important functions that you think your team would like to have. And so for this example, maybe what I'm concerned about are functions like molecular cloning, CRISPR, collaboration, accessibility, user support, and for sure cost. You and your iGEM team may have similar or different functions that you identify as important. But now that I have these, I can actually expand more by adding specific features that I know are probably reasonable for these and also fit the needs of my team. So for instance, if I'm interested for molecular cloning, what particular options I have for these, and what are these discrete features, whether it's actually primer design, whether it's sequence visualization and alignment, or maybe it's actually assembly, manual or automated assemblies. So then once you kind of have an extreme feature list for all of these functions, I think this is a great time to compare your existing options or even new ones. And so for this example, I'm actually using design software that I used to use in the lab myself. Hopefully you all are somewhat familiar with Benchling by now, but when I was around, and these programs are actually still around, I used to use things like Ape, a plasmid editor, LaserGene, and Office Suite software like many of like Microsoft Office or Google Suite. And then what I really like to do is that when I compare individually the functionalities and features for all of these, it was really clear to me which ones would really be impactful and really productive to my overall goal. All right. And now I think that this is really important. I want to kind of segue and transition this into thinking about how does that your biology design software is really going to impact one of your biggest deliverables for the iGEM competition. And so for the most part, I've listed 3 examples here that are probably the most relevant to having a good biology design system and tool. So for instance, for any sequences that your team designs and builds, you're going to need to document them in parts on the iGEM registry. These sequences need to be accurate and really detailed about how you obtain these sequences and what the purpose of the sequence was for. So a great way of doing that is using your biology design software to catalog these parts and have that and hold that sequence information for you. Additionally, you're also going to need to give credit to whoever designed or even made these constructs experimentally. If you have design software that can actually attribute and trace back where these sequences you're working with and who made them, this is going to simplify project attributions tremendously. And finally, the team poster you have to present can often have a lot of different information depending on what stage of the project you came into. So this can include experimental design and even maybe sequence information. So having the ability to really take images of plasmids or your DNA sequence can be super helpful while preparing your poster. And with that, of course, before we get into the demo, I want to give you all a quick review of the Benchling Molecular Biology platform. It's one of our 2 major applications that we offer. Our microbiology suite is really a unified interface that's a leader in sequence visualization and design. There are tons of features that we offer and that we're going to go over today, such as sequence alignments, construct design, and so on. But especially for iGEM teams, we actually have the ability to import parts and sequences from the iGEM registry. We've heard from previous iGEM teams as well that who have used Benchling, that's a great way to unify their tools on a single place, accelerate workflows with cutting edge capabilities, and really establish data transparency for everyone. So now I'm going to actually hand it off to Sierra to give you an outline of what she's going to cover today, and then for her to actually go into the demo. Sierra?
Yeah, thank you, Johnny. So today, like Johnny sort of mentioned, we're going to be going through a workflow, a demo workflow. That's going to start off with actually creating and importing DNA sequences into Benchling. Then we're going to go over editing and annotating those specific sequences. And then finally, as voted from you all in the previous session, we're going to be going over designing constructs for cloning within Benchling. And then we're going to go over aligning those DNA sequences and finally end off with designing CRISPR guide RNAs. So with that, I'm going to, if Johnny, if I could take over the screen share, I'm going to go ahead and move over into Benchling.
Awesome. Okay. I'm going to hit stop sharing, or are you going to? we go.
All right, everyone. And now, so again, welcome to the third installment of the training series for iGEM. And so for the purposes of the recording and to be less distracting, I'll have my camera off for this part of the presentation. So it would be appreciated if everyone would be able to, if everyone could listen and to the demo that I'm going to be walking through and then try it out for themselves on their own computers once the demo is complete, and it will be recorded for your own reference and posted to YouTube after the fact. All right, so in previous sessions, we went over an introduction to benchling and how to master notebook entries. Today, I'm going to be walking you through our molecular biology application and all of its amazing features and tools. To give you a brief refresher of benchling navigation before you start, once you open up benchling, you're going to be seeing an empty workspace such as you see here. This means that I do not have any entries or sequences open at this time. On the left side of the page is the navigation bar that you can see over here. At the top is the jellyfish logo, which when you click on it will bring you to our home page. And from here you can see today's date along with any recently opened entries or sequences. Now below the home button is the projects icon. And within the projects icon, which is indicated by the brief face, clicking on this will bring you to all of the existing projects you have within your Benchling account. And we'll be going back here in a second. Now, next in the line is represented by the eyeglass icon, we have our global search. Here's where you'll be able to search and filter through all of your entries and sequences that you have in Benchling. And I find this tool especially helpful to be able to search through my sequences and applying filters such as being able to search via a specific string of nucleotides. You can also apply other filters such as looking who created your sequence as well. And that can be found through clicking on this filter tab and adding filters from here. Next, below the global search function is the global create button. Now there are a lot of options for what we can choose, such as creating a new project or a new entry, but what we're going to be focusing on today are these bottom options. From this menu, we can create a new DNA sequence, a new amino acid sequence, a new oligo, or a new CRISPR guide RNA. We'll be coming back to the new create new DNA sequence in a second. And next at the bottom, very bottom of our navigation bar is our avatar. This is where you can access your settings. And then finally on the bottom right over here is the help icon. And here's where you can easily access all of our excellent help articles, along with sending a chat to someone here at Benchling if you require any help or need any clarification while you're using the platform. Next, we're going to be going into actually working through that demo workflow that I spoke to in the outline. So going back to the global create button, I'm going to hover over the DNA sequence, and now I'm going to be clicking new DNA sequence. In this new modal that pops up, there are 4 options to choose from. We have input a raw sequence, select a chromosomal region, search an external database, or convert files. So you would use the input raw sequence function if you have a string of nucleotides that you wish to use to create a sequence in Benchling just through a raw input file. You would use the select chromosomal region function if there is a genome you are interested in that you would like to import a specific portion of that chromosome. The third option is to search an external database, which allows you to, as the name implies, search the database where you will, searching through a specific sequence or sequence name. And we're going to be using this feature in a little bit. And then finally, the fourth option allows you to convert files from your computer. And this might be especially helpful if you have any downloaded sequences from your iGEM team members or an iGEM, any other external members that you would like to import into Benchling. So today, we're going to start by using the search external database function, as I said. Now I have another tab open with NCBI. And this is a specific NCBI page which is a very applicable page for what's going on in the world right now for the coronavirus 2 isolate. Now I see there's a lot of information here, but what I need in order to import into Benchling is our accession number. So I'm going to highlight this. I'm going to copy the number, go back to my Benchling page, and paste that into the search bar. I'm then going to search, and what Benchling does is going to search through NCBI and come up with a result for this severe version of the coronavirus. I'm just going to change the name to make it more accessible to myself. I'm going to call this SARS-CoV-2. And I'm also going to double check to make sure that it's saved to the correct project in Benchling, which is this iGEM project I'll be working with today. The next step is going to be actually going ahead and importing this sequence into Benchling. Right now, Benchling is reading the NCBI database and making sure to create that import. Alternatively, another way to import sequences into Benchling is to actually go back to our project. I'm going to click into this specific project that I'll be working with today. I'm going to go to the plus button over here and click Import DNA Sequences. From here, I'm going to go click back over to our external database. But instead of going into NCBI this time, I'm going to go to the iGEM registry that you all have access to. Now from here is basically a list. I've navigated to a list of promoters that are available to you all, and I'm going to select the first one on the list. What I'm going to do here is I'm going to highlight the name. I'm going to copy it through Control C on my computer keyboard. I'll go back to Benchling and redo the search, but this time with a new registry option. Now I can see the correct promoter populated over here, verify the folder is correct that I'm importing into, and then I'll click import. And what Benchling will do is finally load this promoter sequence into Benchling from the iGEM registry. Now going back to our original SARS-CoV-2 sequence, you can see a variety of different annotations that are appearing in our screen. To expand this workspace or collapse it, we can actually click this button over here, Split Workspace. This will allow me to split my view from sequence map to a linear map, looking at the metadata or description tabs, and then we can just undo this by re-clicking Split Workspace. Now the available tabs in the sequence map, which allows you to see the sequence and any annotations or primaries associated to the sequence. The linear map, which is over here, which allows you to see your sequence in a linear format. A description, this tab over here, which will be automatically filled in with information from the database where you imported your sequence from, along with where you can actually @mention a user on this sequence. So for example, I'm going to @mention Johnny to this sequence. Maybe for your example, you would like one of your iGEM team members to be able to maybe take a note of the sequence that you've imported, and maybe you want them to collaborate with you, or any of those reasons would be great ways in which @mentioning a user would be super helpful in this case. And then finally, we have our metadata tab, which is where you can see the author or person who imported the sequence, the project where it's saved to, and when it was created. These tabs can also be moved around, so in order to maximize your real estate, as we call. So right now, I'm going to unsplit my workspace so everyone's able to see my SARS-CoV-2 sequence. And actually, another hidden trick for Benchling is to collapse this projects menu. I'm going to re-click on the briefcase to really maximize that real estate. Now, to interact with the sequence, you can click around at any point throughout the sequence, such as over here. I can bounce around, and you might notice that when I highlight a portion of my sequence, a couple of the different parameters change down here. So I'd like you all to take a look down here, and then when I do highlight different parts of the sequence, what's going to change is the start and end. Redo that. The start and end is going to change. The length of the sequence that I have highlighted, the GC percentage will change based on what I have highlighted, as well as the melting temperature. Now let's say I would like to add bases to my sequence. I can simply click on a part of the sequence where I would like to add bases and just start typing some nucleotides, such as AT, 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 for example. I can then click enter, and what will be entered in is those bases that I had typed in. I can also right-click on a part of the sequence where I would like to input bases, click, right-click, insert bases, and that same modal will appear. I'm just going to exit out of here right now because I've already inserted some bases into my sequence. To remove bases, I can highlight part of the sequence I want to remove and click the delete button on my keyboard. So I'm just clicking that delete button, and those bases will then be deleted. Or I can highlight the part of the sequence I wish to delete, right-click, and delete bases from here. But let's say I made a mistake and I didn't actually want to delete those bases. You can undo in Benchling through clicking Command Z or Control Z on your keyboard to undo the last edit, such as what I'll be doing right here. Oops, there we go. Click that again, undo. All right, so other ways you can adjust views for your sequences is to locate the gear icon on the top right of your screen, which is over here where my mouse is. From here is where you can toggle on or off various aspects of your sequence, such as toggling on or off the annotations, any primers, any primer bases, cut sites, open reading frames. I can toggle those on and off so you can see those from here, and more. So this can be really handy if you would like a cleaned up version of your sequence. Maybe you only want the bases, or maybe you want all of the options possible just so you can see every attribute of your sequence. I'll leave those on as it is for now. Another way to adjust your sequence or analyze your sequence is to highlight and create a translation. So I'm going to be highlighting this portion of my sequence over here. And to create a translation, I can right-click, go down to Create Translation. Now Benchling gives me the option to either create a translation on the forward or reverse sequence. I'm going to select forward for the purposes of the demonstration today. So what you will see here is the amino acid string that is created from this string of nucleotides. Now, on the right side of my screen is where you can see a variety of different icons and applications or parts of the molecular biology suite that we'll be talking with today. To start off, we'll be talking about the annotations icon over here. Clicking on this, the flag icon, will bring up a new menu which lists all of the annotations currently on this sequence. Now let's pretend we want to create a new annotation on our sequence. Maybe there's a site of interest that I'd like to denote, or something I would like to perform an experiment on. I can highlight a portion of my sequence, and then I can click New Annotation over here. What's going to pop down in my menu over here, I can give the annotation a name. For example, I'll call this New Annotation. I can give it a type if I wish. Benchling will automatically tell me the position that is currently highlighted in my sequence where this annotation will end up. I can give the annotation a new color if I want. I'll just leave it as the automatic green that is selected. And here I can also designate either if the annotation is on the forward or reverse strands. I'll leave it on the forward strands for my demonstration today. Then I'll go ahead and save that annotation to my sequence, and now this new annotation will be saved. Now I'm going to click out of my annotations menu, and let's say I'd like to be able to easily highlight this new annotation that I made, all of these sequences, and I would like to copy them to a new menu. To do so, what I can do is I can just click on that annotation, and that eventually will automatically highlight all of the bases that are contained within the annotation that I just created. Now that we've talked through the basics of our molecular biology tool, we are now going to walk through the top voted topic from last time, which was how to design constructs within Benchling. To demonstrate how to do this with you all, I'm going to be taking one of the genes from the SARS-CoV-2 genome and Gibson cloning that into a plasmid PET31B, along with fusing that SARS-CoV-2 gene with a GFP gene. This may be a similar experiment to what you were planning with your iGEM projects, or maybe you might be using the COVID genome as well for your projects. So the first step in construct design would be PCRing out a gene of interest from our SARS-CoV-2 genome. In order to run a PCR, we first need to outline two items. The first, the exact gene we are looking to clone, and the second, designing primers in order to run the PCR reaction. I'm going to choose this ORF1 leader protein sequence as the gene of interest for my cloning experiment. And again, you can see when I click on this specific annotation, all of the bases of interest will be highlighted. Now that we have recognized our gene, let's design some primers. So within Benchling, we have a primer design modal. I'll be clicking on that over here. It's indicated by the double arrows. From here, we can click on Create Primers, and we can either choose using Benchling's manual tool or using our primer wizard. I'm going to be showing you all how to use our manual tool today. So I'll click on that. Now, this is a time where I would want to resplit my workspace so I can both see my sequence map and design primers tool at the same time. So I'll go ahead and do that. And from here, because I want to see our sequence map, I'm going to drag and drop my design primers tool over to the right side of my screen. So from this top dropdown over here, because I'm looking to run a PCR reaction, I'm actually going to want to design a primer pair instead of a single primer. Next, I'm going to want to highlight the portion of the sequence I would like to make my forward primer. So from here, I'm just going to select about 20 nucleotides, which is a normal amount for a primer. Let's say I'll choose these 20 nucleotides for my forward primer. I'm then going to jump over to my Set from Selection button over here, and I'll designate this as my forward primer. And Benchling will auto-populate those bases into this bar over here. Now I'll scroll down to the end of my ORF1 leader protein gene. There we go. Now that we've at the end, I'm going to now designate my reverse primer bases. So I'll go ahead and highlight again about 20 bases in order to run my PCR reaction. So there we go. We have about 20 bases selected. I'm going to go back to Set from Selection and designate this as my reverse primer. Benchling will tell you the 3' location of the primer along with the option to select maybe insert a restriction enzyme cut site or to create an overhang. But because we are using Gibson cloning, we're going to leave our primers as is. I would also like to note that the TMs of these primers may not be within 5 degrees of each other, which is usually a best practice, but for the purposes of today's demonstration, I'm going to be leaving these primers as is. And we'd like to note that every iGEM team should have a standard for primer guidelines when they are designing their primers for their experiments. Also within our primers tool, we can check for secondary structures if that is a concern of mine. So I'll go ahead and click this button to check for any secondary structures that might form. And then the final step is to give these primers a name and save them to a folder. So I'm going to give the forward primer, I'm going to just call this forward. And then I'll give my reverse primer, I'll call that reverse. I'll make sure to indicate the correct folders I would like to save my primers to. This one has the correct folder, but this one I need to choose a folder for, and I'll choose my iGEM project that I'll be working with today. Make sure to click select. And then I can save this primer pair to my sequence. Benchling will then load and then notify me that the primers were successfully linked to my sequence. So I'm going to actually unsplit my workspace so we're all able to see these primers in action a little better. So you can see my forward primer up here, and if I scroll down to the end of my gene, I can see the reverse primer indicated here. Now a common practice next is to actually, what we're going to need to do is to PCR out or create a PCR product from our primers that we had just created. So I'm going to go ahead and click this Create PCR button in order to create this product. Next is what's going to populate is a new menu will allow you to customize what exactly gets copied over from our original sequence to our new sequence, such as annotations, tags, and any other items that are listed here. I'm going to leave these selections as is, these are all items that are of interest to me, and then I'm going to click copy. Make sure my project is verified, and then click select. And then what Benchling will do is to create this new PCR product. I'll scroll down, we have both of our primers, and this looks great to me. But next is a common practice to verify that our PCR product was successfully created is to go through a restriction digest with our product. Now we can, within Benchling, I'm just gonna make sure that restriction enzymes cut sites are indicated on my sequence, which is correct. And so within Benchling, we can actually simulate a digest and calculate the fragment size so that when you are in a lab, you're able to have an expected result for your digest and electrophoresis gel run. To select a restricted enzyme, I'm going to go to our digest tool indicated by our scissors icon over here. From here, I have a variety of options and enzyme lists, such as searching through all commercial enzymes in Benchling or by a specific vendor, such as NEV. I can also display enzymes that cut at specific points in the sequence. I'm going to make sure to select single cutters so that I can properly select an enzyme to verify my PCR product, and I will search by 1. And what's going to populate in this list over here are all enzymes that cut my sequence once. Now, if I scroll down a little bit, I can see that this specific enzyme, BSM1, cuts my sequence once and also is roughly in the middle of the sequence. And I think this would be an excellent candidate in order to verify my PCR product. So I'm going to go ahead and click on this sequence to make sure it is, or its enzyme, excuse me, to make sure it's highlighted on my sequence. Now what's available on the bottom of my screen is that I can now have the option to run a digest in Benchling. So I'm going to go ahead and go through and actually run this digest. And what's going to appear on a new page is the start and end length of my two fragments that are then created from this restriction enzyme digest. I can see the optimal buffers that this enzyme works best in, along with any overhangs as well. And then there's a new tab that also pops up on this page, which is the virtual digest. Now, I think this is a really awesome part of Benchling, which allows you to show a visual representation of what my gel might look like after I run this specific restriction enzyme digest. I'm able to select from a variety of ladders, such as a 1 kb ladder, or you can input other ladders into Benchling. And then what you'll see over here is that I can see both of my two fragments. They're kind of close together, but you can see both of my fragments indicated over here and how big they are by highlighting. And this is really handy if you are running gels and running lots of digests in the lab. I think this would be very useful in order to actually see what your expected result would be before actually running and going ahead with your digest. So now that my PCR product is restriction enzyme verified, let's go into the actual construction of our new plasmid. So I'll hop back to our sequence map. So like I said before, so let's assume you're working with an iGEM team and you want to be able to fuse this specific gene from the SARS-CoV-2 genome with GFP and actually clone it into a specific vector. And I'm going to be walking you through this process with Benchling. So with that, I'm going to be going back to my projects and project inventory. So you might see that I have a plasmid backbone that's already been linearized in Benchling, the PET31B. I'll open that up. And then I'll also want to open up my GFP gene that I've also already imported into Benchling. So in terms of assembly, Benchling offers a variety of options, and I can find all of those options by going to the bottom of my screen and hovering over and clicking on Assembly Wizard. I could then choose the option to create a new assembly, and Benchling allows you to pick from 3 options: Digest and Ligate, Gibson, and Golden Gate. If there are other types of assembly methods you are working with that might not be on this list, you can always model these types of assembly through copy and pasting portions of sequences together and designing primers accordingly, and inserting any restriction enzyme sites into your sequence if that is what the type of cloning method you are working with. But for today, I'm going to be selecting Gibson assembly. Gibson assembly in Benchling is a great option because all you need to tell Benchling is what your backbone sequence is along with any fragments, and Benchling will do all of the work to design your homology primers for your experiment. I think this is super handy in order to ease your design, use, and everything like that. It makes Benchling a really great tool. So I'm going to click Start, and Benchling will then prompt me on the bottom over here to designate a backbone and fragments for the assembly. First, I'm going to go over to my backbone, the PET31B sequence. I'll then click into that, and I'm going to Command A on my keyboard to highlight the entire sequence. I'll then select backbone and set the fragment that I have highlighted as my backbone. Next, I'm going to want to click on the Insert tab. I'll go over to my gene that I cloned out of my SARS-CoV-2 genome. I'll click into that specific sequence again, Command A to select all, and then I'll click Select Fragment to designate this as my first fragment. Now I would like to also add in my GFP to create a GFP fusion. So I'll click the plus button over here, and then the new Insert tab will be open. And I'll hop over to my GFP gene over here. I'll click into that sequence, Command A to select all, and then set as a fragment. Now I have my backbone and both of my inserts indicated in Benchling. I'll then change the name of my untitled assembly. I'll give it a name such as PET31B. SARS-CoV-2-GFP, just for an example. I will then click Assemble. And what Benchling is going to do, I'm going to first indicate to make sure the sequence and primers are saved to the correct folders. I'll then create, and what Benchling will do is it's going to run this assembly and provide me all of the primers needed to run the assembly for myself in the lab. Now the final step to verify my assembly, I'm going to click over to the Assembly tab, and then if everything looks good to me, I can look at all my primers, make sure everything checks out. It looks great to me for this experiment, and then I'll click Finalize. And Benchling will then, once you click Finalize, finalize your sequence, and now we have another new option that we didn't see before, is to look at our plasmid map. Now from over here, we can see our plasmid map and our entire construct that we just created. We can see our GFP gene. Right over here is our ORF1 leader protein genes. And then we can see our backbone that has ampicillin and LAC1 genes as well. Now that we have created our construct in Benchling, it is best practice once you create a construct is to sequence verify it so that you're sure what you have created matches what was expected. A great way to do that in Benchling is to utilize our alignments tool. I've already created some sequencing files for us to use to create an alignment, but from here is where you would send out your construct for sequencing and then receive those back from wherever you were sending your construct for sequencing. First, I'm going to go into our alignments tool, which is indicated by these multiple bars over here. I'll click that and then select Create New Alignment. Now, with a new menu will pop up, which will allow me to search for a sequence in Benchling, maybe if I would like to align for that. But for today, I'm going to choose files from my computer. I have 3 files that I'll be inserting in for my alignment. And those will be populated over here. I then want to make sure my PET31B construct, new construct, is designated as the template. I can change the template by clicking on and off that specific sequence, but I want to make sure to designate that as the template for our alignment today. Once I have all my sequence files inserted into Benchling and I have my correct template designated, I'll go ahead and create my alignment. Benchling will then load and calculate our alignment from our files that we had inserted. And then what will appear over here is our template sequence at the top and all 3 of our sequence files down here. I can scroll through my alignment click on my keyboard. I'll scroll through and see all of the different peaks that we have over here. And if you hover over a peak, Benchling actually analyzes the quality of what peaks were generated for your sequencing file, and I can see the quality from there. So scrolling over, another way to go more quickly throughout your alignment, as this is sort of a long alignment, I can click on the orange bar down here and click and drag to a specific part of my sequence. Now, if I go to the gear icon, similar to our sequence view, if I go to the gear icon up here, this is where I can turn on and off any translations, annotations, any primers that are on my sequence, any other qualities of my annotation. Now, this looks like a really great sequencing result to me, but I do see these 2 red bars over here. Now, these red bars indicate a mismatch. In order to hop through mismatches fairly quickly in Benchling, specifically in our alignment tool, I can click these left and right arrows. So I'll click the right arrow, and what Benchling will do is jump right to our mismatch over here. It looks like we have 3 bases that are missing from our template that were found in my sequence file over here. Maybe this was a planned sequence insertions based on our primers, or whatever reason that might be, or maybe this is actually a red flag for me that maybe my construct or that I had sent for sequencing maybe has some mismatches. And maybe I would like to indicate this to one of my fellow teammates in my iGEM team. What I can do is I will select this space over here, and I'm going to click Add Comment. And here is where I can add a comment, such as I'll say mismatch, maybe with a question mark, or whatever comment that might be, and I can actually save that to our alignment, maybe to reference back at a later point. I'll then click Save. And then I can actually, to show you other mismatches that I have within Benchling, Let's say I'd like to jump back over to this portion of our sequence, and just for the purposes of today's demonstration, I will insert some new bases over here, such as AT. Just to show you a little cleaner of what it would look like is that these 4 bars will indicate on our template that they're not present, and that on our sequence file over here, we can see these new bases inserted and they're highlighted in red. What you might notice up here are votes, which is indicated in gray boxes. The votes indicate the number of sequences that match together. So for example, we have our 1 sequence file, and that indicates 1 vote, or 2 votes, or etc., and how that will affect your sequence. You can also turn these off again by going to our gear icon on the top right and toggling on or off the votes. Now that we have sequence verified our construct, maybe all the annotations, or not annotations, I should say, all the mismatches we did find, we may have expected them, or they might code for an amino acid that will not affect our construct. We can say our sequence is finally verified. Now I'll hop back over to our sequence map, or plasmid map actually, so we can all see a great view of that. Now we have finally moved on and created our construct, and let's talk about actually our CRISPR tool and the last time that we have today. So the CRISPR tool was another very popular topic from our last webinar, and to do so, I'm going to go back to our original SARS-CoV-2 gene. I'll unclick my primers tool, and let's take a look at what we have over here. So let's say I would like to create a guide RNA for a gene editing on a specific gene, maybe this ORF1 gene in my sequence. I'm going to select the gene we have been working with today, like I said, and I did so by clicking on that annotation. Then clicking on our CRISPR icon indicated by the target icon over here, I can select either, my options are to design and analyze guide RNAs or to design a homology recombination template. Today, I'm going to be choosing design and analyzing guides. A new menu will then pop up, which will allow me to choose different design parameters, such as the type of the design you are looking to do, if you were looking to do single guide or paired guides, or guides for base editing as deemed by this paper, the length of the guide RNA that we'll be producing, the genome to design our guides against, and if I would like to indicate a different PAM sequence than what is automatically selected. I'm going to leave all the parameters as is in order to do our guide design today, but you are more than welcome to adjust them for how you are running, if you are running any CRISPR experiments with your projects. I'll then click finish, and Benchling is going to verify that the correct region of interest that I'll be working with, and I'll verify that this is indeed correct, and I'll click create. Benchling will then calculate all of the guide RNAs associated with the specific parameters that I had defined in the previous menu. I'm going to resplit my workspace so I can see both my design CRISPR module and my sequence map at the same time. I'll also collapse my project menu over here so we can see a little better. So below are all of the guide RNAs that were calculated, the guide RNAs possible that were calculated for the sequence selected. From here, I can either sort by on or off target score, depending on what sequences or what is necessary for your projects. I'm going to sort by on target scores today, and I'm just going to select, for the purposes of this experiment, I'll say this specific guide RNA looks best for me. From here, I can save this to my sequence or assemble it into my sequence as well. I'll just be saving it to my sequence today, and I'm going to make sure to indicate the correct folder I'll be working with. I'll then click save, and what's going to be saved to my sequence is this specific guide RNA. Even though that's a brief overview of our CRISPR tool, for the sake of time, you can also assemble CRISPR guides into commonly used CRISPR plasmid sequences, but we're not going to be going over all of that specifically on how to do so today. I'm going to once again unsplit my workspace so we can see this specific guide RNA. And then finally, what I'm going to be touching on today is our info icon down here. Clicking on that will bring me to a new menu, which will tell you the name of my sequence, and this is where I can change that if I do wish. It will show you the exact folder where my sequence is stored, the topology of my sequence. I know this is the linear genome. The start index, meaning where the sequence will begin. For this sequence, it's at position 1. I can lock or unlock my sequence from any edits. And then when it was created, the length of my sequence, and if I had any updates to make to this information, I would be able to click this update information button. But what I would really like to touch on today is our export data option. Benchling will allow you to export your sequence data through any of these file formats. So let's say you're working with an external collaborator who may not have access to Benchling, or for whatever reason you would like to export your sequence file. This is where you can do so either, like I said, through any of these file types such as GenBake or FASTA files. From here is where I can archive, and then after I archive, I can delete my sequence. Maybe it's a mistaken sequence that I might have imported into Benchling for whatever reason. So with that, that sums up our exploration today into the molecular biology suite in Benchling and how it can be an excellent source in order to create constructs for your iGEM projects. As always, you can reach out to us in Benchling through clicking the question mark icon on the bottom right, and then going to the chat feature. And now I'm going to be handing it back to Johnny to finish off the rest of our webinar today.
Awesome. So hopefully everyone can see everything. Awesome. And so I know that that's a lot of information, and Sarah gave an amazing workflow for our biology suite today. I want to introduce a small caveat. Hopefully I've addressed this in the chat, but if you do plan to do any kind of COVID-19 work for your iGEM project, please make sure to check the iGEM guidelines on the iGEM website and make sure that you take a look at the safety forms that you need to fill out if you plan to pursue those types of research projects. For the purposes of this demonstration, Benchling just wants to show you how you can integrate those particular workflows, but still up to your iGEM team if that's something you choose to undergo. And so quickly for that, I just want to give some big takeaways from Sierra's great demo is that, and so first of all, in terms of, remember that you can essentially just collate DNA sequences from a variety of sources, whether that's uploading sequences, whether that's importing it from NCBI or your iGEM registry. Make sure to take advantage of this particular feature and be able to easily take sequences into Benchling and even out of them. Second, make sure that for those of you who understand the sequence parts and annotations that you need for your iGEM team, you can customize these across your entire team, making sure that each member is using the right promoter, the right sequence at all times for every experiment. And so be sure to take advantage of that. And third, one of Sarah's big parts in this particular presentation is how cloning can be done. And although we've shown you a way to do it through our assembly wizard, you can always be doing cloning manually through our really easy copy and paste options, depending on what type of cloning method you would like to try. And number 4, you can see that you can align and analyze sequencing files very easily, which can really help determine the next steps within your experiment. Like Sierra mentioned, whether it's a mistake in the sequencing or whether or not you need to reinvent your particular cloning strategy, you can easily tell by compiling all of these particular tools onto Benchling and essentially your sequence files as well. And then lastly, designing and scoring CRISPR guide RNAs can be a very difficult challenge, and also understanding what's the type of software to use can also be really, really hard to assess. Benchling has a built-in CRISPR tool that you literally saw you could design CRISPR RNAs in almost seconds. And so if that's something you plan to partake into your particular iGEM project, please please think of Benchling when you're trying to use that. All right. And so I know that this is and now we're getting ready to wrap up, but I actually want to hear a little bit more from you all about what features you really really interested in for today. And so essentially, I'm just going to give you guys a really quick poll about what was your favorite feature today. If you guys have a chance just to just only unfortunately have to choose one, which one would it be? So we'll give a little time for everyone to kind of put in their responses, and we'd love to hear everyone's feedback, especially so we know how what iGEM teams are thinking about the most, and also what aspects of Benchling we could improve even more. Okay, ending the poll now. And I'm going to share these results with you all. I'm glad to hear that the majority of you guys all really liked essentially all the features that we had to share with you all today. Sierra spent an amazing amount of effort and time to really cater this demo for you all and for the use cases that you guys all typically use. Okay, so now I think I want to move on to make sure we have some dedicated time for a question and answer session. Um, so really quickly, we're gonna be checking the Q&A poll right now, and we're gonna see if there's any particular audience questions that you think that you have. All right, so really quickly, I'm just gonna exit this. Sorry, having a small time seeing. So one question asked is, why was there a mismatch? Does that mean the construct would not work experimentally? How are we meant to deal and solve with mismatches if we find any? Yeah, so for those of you who, I think this is relating to the sequencing tool. So if you do find a mismatch within your particular design sequence, we are assuming that you experimentally created that particular sequence. And then when you send it for sequencing from another company or another provider, you found out that when you realigned it to your desired sequence, it did not fundamentally match everything. So in that particular way, this is the part of troubleshooting in science where you have to figure out what went wrong or what part do you need to reiterate. This could be a number of things, whether it's reiterating on your cloning strategy, or maybe it's your particular construct that you sent off for sequencing. Maybe you need to send off a different clone. Maybe you need to send off or so forth. And so that's the part where benchling really helps with the science. But unfortunately, we can't interpret the science for you. We can only help you visualize it easier. Is there anything you'd like to add to that, Sierra?
No, that was a perfect explanation, Johnny. Yeah, absolutely.
Okay, awesome. One person asked, does Benchling have a support team to contact for troubleshooting experiments and design? Do you want to take that, Sierra?
Yeah, absolutely. Sure. Yeah, we have our support team on call. So whenever you submit a ticket, if you click that chat option, we're on call to answer benchling specific questions. We're not trained to exactly answer experiment specific or design specific questions that you might have, but we can try our best to help.
Mhm. Yeah. And like Sierra is one of many who answers particular science questions, she also has a science background, so it only makes her expertise even better. But of course, we may not know your particular science as well as you do. Okay. One quick question is, how accurate is the electrophoresis simulated in benchling with actual lab electrophoresis? Yeah, I can take a stab at this, but in my experience running also a lot of lab electrophoresis gels, it can also really depend on your particular sequence, how it runs. The virtual digest that Sarah showed is an approximation of what it should look like. And it's just to give you a base of reference. Of course, with any gel that you run, you want to have the right controls, whether it's the template sequence or a sequence of another comparable size to what you're measuring. So I think it's always important to have these particular controls, but benchling, unfortunately, it can't provide every single piece of insight for that. All right. And then another question, I think one of our last questions is it possible to import Snapgene files with annotations? I think to my knowledge, you are able to export particular Snapgene files out as either a GenBank or a FASTA file. But unfortunately, the way you export them out from Snapgene, they wouldn't particularly have annotations that you need. One thing that we do have at Benchling is that we have something that Sierra didn't get to cover is annotation libraries. If you have a specific set of annotations that you want to use for every sequence, you can actually create a custom annotation library, and you can make sure that it's applied across all of the sequence that you generate. Yeah, Sierra, do you want to explain that a little bit more, or is there anything I missed about that? Mm-hmm.
No, yeah, absolutely. Yeah, annotations, I didn't have enough time to touch on it today, but basically how they function in Benchling, you can click and auto annotate on your annotations tab, and that will be able to source and match every single sequence that you have mapped out in your annotation library and match that to a specific sequence, whatever that might be. If you're such as GFP, if you often are using GFP and you would like to indicate it in your constructs, that would be a great example.
Awesome. And now I think, unfortunately, we're getting time to wrap up. I just want to take a few minutes really quickly, just to like one extra minute to show you guys all a little something about, I want to show you that Benchling has been really dedicated for helping out iGEM teams worldwide help with their projects and their research. And so essentially, we have been involved in a number of activities, whether it's webinars, whether it's workshops, or even essentially panels. We really like to hear actually from you all about if there are opportunities locally with your iGEM team or wherever you are doing your iGEM projects, if there's an opportunity to help out. And if so, feel free to reach out to Johnny at benchling.com. Or also, feel free to follow us on social media, and you can even submit ideas and look for news and events even there. But yeah, other than that, we really have enjoyed preparing this iGEM webinar series for you all today. Hopefully we'll see you all soon in other capacities as well. So thanks everyone for tuning in today, and bye everyone.